Hi everyone, this is Jahnavi's Biology. Welcome to my channel. Today we are going to learn about dihybrid cross. In this dihybrid cross, we are going to cross between two plants having two different traits. The plant here we are going to use is pea plant. This pea plant is experimental tool of Gregor Mendel, who is called as father of genetics. Here, the pea plant, we are going to take two different parents with the two different traits. The parents we are going to take as capital Y, capital Y, capital R, capital R, small y, small y, small r, small r. In this, we are going to get yellow and round for capital Y, capital Y, capital R, and green and wrinkled for small y, small r, small r. From these are homozygous in nature, so we are going to get only one type of gametes from both the plants. In this, we are going to get for yellow round capital Y, capital R, and for green and wrinkle, we are going to get small y and small r. From these two gametes, for F1 generation, we are going to get capital Y, capital R, small r as the heterozygous plant, which is our first generation progeny. This is yellow and round in nature in heterozygous condition also. So yellow and round are taken as or considered as dominant nature traits, and the green and wrinkled are taken as recessive nature traits. In F1 generation, all the plants which were grown in F1 generation, all the plants will be getting capital Y, capital R, smaller genotype only, and it will get the phenotype yellow and round. These yellow and round are the color and the shape of the seeds. Now, if we want to get the F2 generation plants, we should self between the two F1 generation plants. Means we want to self between the two heterozygous yellow and round plants to get the F2 generation progeny. We will take the parents of F2 generation as capital Y, capital R, small r, and capital Y, capital R, small r. It is so different from the monohybrid cross. In this, two different heterozygosity nature traits are being there. So, for this, we are going to use a formula to get the number of gametes. It is 2 power n. Here, n refers to number of heterozygotes. Here, in each parent, we are going to get two heterozygotes. Means, in first parent, capital Y, capital R, small r, we are going to get capital Y, small y, a single heterozygote, and capital R, small r, another heterozygote. So, here in each parent, we are going to get two heterozygotes. So, we are getting four gametes from both the parents. In the first parent, we are going to get capital Y, capital R, capital Y, small y, capital R, small y, small r. These are the four gametes that we get from the first parent, and from second parent also it is having two heterozygotes, and so it is getting four gametes from this. So these four gametes are also same as the first parent. They are capital Y, capital R, small y, capital R, small y, small r, and capital Y, small r. As there are totally 8 gametes formed, we are going to use Punnett square method. Punnett square method, we should use Punnett square method because to know the maximum possible progenies formed from these 8 gametes, which we are considered as F2 generation progeny. Let's draw the Punnett square to know the maximum progeny that we will get in the F2 generation of the dihybrid cross of pea plant. In F2 generation, we are getting 4 gametes from each plant, so that we are going to use Punnett square method. In this Punnett square method, as 4 gametes are given from both the parents, we are getting 16 progeny. As you are seeing in the video, we are using Punnett square method to take the maximum progeny we are getting from F2 generation. That capital Y, capital R, capital R, capital Y, capital R, small r, capital Y, small y, capital R, capital Y, small y, capital R, small r, capital Y, capital R, small r, capital Y, small r, capital Y, small y, capital R, capital Y, small y, small r, and capital Y, small y, capital R, capital Y, small y, capital R, small y, capital R, small y, capital R, small r, and capital Y, small y, capital R, small r, capital Y, small y, small r, small y, capital R, small r, small y, smaller. These are the 16 plants we are getting as the progeny of the F2 generation of Dihybrid Cross. In these also, we are going to get phenotypic ratio and xenotypic ratio. In this, the phenotypic ratio will be being on the basis of the morphological characters which are seen from our eyes, by our eyes. It is in the ratio 9:3:1. And genotypic ratio will be somewhat complicated, but we are going to understand in the new method. The xenotypic ratio is 9:3:1. To know the genotypic ratio, we already know the phenotypic ratio. To know the xenotypic ratio, I am using method. See carefully that I am only writing the homozygous yellow, heterozygous yellow, and homozygous green characters, and as these are of 3 types and the green is of 1 type and the yellow is of 2 types, there is a possibility of these capital Y to join with capital R, small r, and the capital Y, small y, both also have the chances to combine with capital R, small r, and small r. So, as a result, we are getting the types by combining the both capital Y, and capital R, we are getting 1 type of the progeny we got in the F2 generation. These are capital Y, capital R, capital Y, capital R, small r, capital Y, small r, capital Y, small y, capital R, capital Y, small y, capital R, small r, capital Y, small y, small r, and small y, capital R, small y, capital R, small r, small y, small r. These are the progenies we are getting from the F2 generation. From these capital Y, the probability is for capital Y is 1 by 4, and for capital Y, small y is 2 by 4, small y is 1 by 4. For capital R is 1 by 4, capital R, small r is 2 by 4, small r is 1 by 4. For all these below also we are going to get same ratios. So by these ratios, on multiplying the ratios, we are going to get the ratio of the progeny in the right what we wrote those. In the ratios, we are going to get the ratio of the progeny in the right what we wrote those. For the first one, we are going to get 1 by 16 on multiplying, and for the second one 2 by 16, third one 1 by 16, fourth one 2 by 16, fifth one 4 by 16, sixth one 2 by 16, and the seventh one 1 by 16, eighth one 2 by 16, ninth one 1 by 16. Now, from this, we will get to know the genotypic ratio of the F2 generation of the Dihybrid Cross. It will be being this as, it will be written as the ratio of the ratios of all the progenies what we get from the F2 generation. It will be in the ratio 1:2:1:2:4:2:1:2:1. It is so easy if you understand the basics of the Dihybrid Cross. On this, the questions will be given in the NEET examination. In the Dihybrid Cross, we are using 2 different traits to know the number of phenotypes, number of genotypes, and the number of gametes formed. We have different types of formulas. The first formula is for types of gametes, we are using the formula 2 power n as before we used. N represents the heterozygotes And the type of phenotypes, the formula is also 2 power n. For the type of genotypes, the formula will be 3 power n, and the number of zygotes formed in this will be given as G square. Here G denotes the gametes, means the number of zygotes formula will be 2 power n whole square. In all the above formulas, the n represents the number of heterozygotes in the genotype. These formulas are used in the NEET examination. These formulas are very useful to know the number of gametes, number of genotypes, number of phenotypes very easily. Thank you for watching this video till the end. Thank you very much.